for Health BRIN, the Honorable Ibu Elizabeth Farah Novita Kotrier, SSE PhD, Head of the Eijkman Research Center for Molecular Biology BRIN, our distinguished speaker, Professor Shiro Suetsugu, PhD, Professor in Molecular Medicine and Cell Biology at the Division of Biological Science, Nara Institute of Science and Technology, Japan. Konnichiwa. Our distinguished moderator, Dr. Yudi Nugraha, SSE and Biomed, researcher at the Eijkman Research Center for Molecular Biology, BRIN, and all distinguished guests, researchers, and all participants joining us both in person and online. Welcome to today's scientific lecture organized by the Eijkman Research Center for Molecular Biology Research Organization for Health BRIN, entitled Cellular, Protrusion, and Extracellular Vesicle for Delivery of Bioactive Molecules Between Cells. This scientific lecture provides an excellent opportunity to exchange knowledge and insight on the latest developments in molecular and structural biology, particularly cellular protrusion, extracellular vesicles, and their role in intercellular communication. Ladies and gentlemen, please rise for singing the Indonesian national anthem, Indonesia Raya. Please be seated. Thank you. Ladies and gentlemen, it is our honor to invite Professor Indi Darmayanti, Chairwoman of the Research Organization for Health BRIN, to deliver her opening remarks through video messaging. To Prof. Indi, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Salam sejahtera bagi kita semuanya. Distinguished Professor Shiro Suetsugu from the Nara Institute of Science and Technology, Japan. Dr. Elizabeth Farah Novita Kotrier, Head of the Eijkman Research Center for Molecular Biology. Honorable Head of the All Research Center under the Research Organization for Health BRIN. Distinguished colleagues, researchers, academics, students, and all participants joining us both in person and online. It is my great pleasure to welcome you to this scientific seminar organized by the Ekman Research Center for Molecular Biology BRINs. Today's seminar addresses a fascinating and highly relevant topic, cellular protrusions and extracellular vesicles for delivery bioactive molecular between cells. Cell-to-cell communication is fundamental to almost every biological process. Understanding how cells communicate, remodel their membranes, and transfer bioactive molecule provides important insight to physiological process as well as the mechanism underlying disease. This topic is particularly relevant research, including infection, immunology, cancer, tissue regenerations, and the development of the innovative therapeutic and drug delivery strategies. We are very honored to have the Professor Shiro Sugu with us today. His extensive contributions to the study of the membrane remodeling, actin, cytoskeletons, dynamics, cellular protrusions, and extracellular vesicles have significantly advanced our understanding of the molecular mechanism underlying cellular communication. I would like to emphasize that our collaboration with the NARA Institute of Science and Technology, NICE, has been very close and long-standing. NICE has been an important strategic partner for BRIN in advancing molecular and biomedical research. Most recently, this collaboration has been further strengthened when I had the honor, together with the NICE, to sign a cooperation agreement for joint research under the multi-year GST Nexus funding program, which is also led by Dr. Yudi. This marks an important milestone in deepening our scientific partnership and expanding impactful in that this seminar will not only enrich our scientific knowledge but also provide an opportunity for further strengthen collaboration and scientific network between BRIN and international network institution, particularly NICE. I would like to thank Professor Sugu for kindly sharing his expertise with us. And I also appreciate the Ekman Research Center for Molecular Biology, all heads of the research center under the Research Organization for Health, and everyone involved in organizing this important scientific event. I hope that all participants will take full advantage of this opportunity to learn, exchange ideas, and explore possibilities for future research collaboration. With that, I would like to officially welcome you all today seminar. Thank you, and I wish you a productive and inspiring scientific discussion. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Indi, for your inspiring remarks. Next, we will continue with the second opening remarks. Ibu Elizabeth Farah Novita Cotrier, head of the Ekman Research Center for Molecular Biology BRIN, also through the video messaging. Ibu Farah, time is yours.
Good afternoon, everyone. Distinguished Professor Shiro Suetsugu from Nara Institute of Science and Technology, Japan. Professor Dr. Hewan, NLP Indi Darmayanti, Head of the Research Organization for Health, BRIN. Honorable heads of all research centers under the Research Organization for Health, BRIN. Distinguished colleagues, researchers, academics, students, and all participants joining us both in person and online. First of all, on behalf of the Ekman Research Center for Molecular Biology, I would like to warmly welcome all of you to this seminar. It is a great honor for us to have Professor Shiro Suetsugu as our distinguished speaker today. We are truly grateful for his willingness to share his expertise and scientific insights with the research community in Indonesia. The topic of today's seminar, cellular protrusions and extracellular vehicles for delivery of bioactive molecules between cells, is highly relevant to our collective efforts to deepen the understanding of molecular and cellular communication mechanisms. The extracellular vesicles and cellular membrane dynamics are now widely recognized as key components in intercellular communication, enabling the transfer of proteins, lipids, nucleic acids, and other bioactive molecules. These processes are fundamental not only for normal cellular function, but also for understanding diseases, mechanisms, and developing novel biomedical applications. I would also like to highlight that at the Eichmann Research Center for Molecular Biology, our research focus is strongly centered on molecular mechanisms, particularly in the cellular and subcellular context. We are deeply interested in how molecular processes regulate cellular behavior, interaction, and function at the mechanistic level. In this regard, the field developed by Professor Suetsugu, especially his work on cellular protrusions, actin dynamics, and membrane remodeling, aligns very closely with our research direction at Eichmann Research Center for Molecular Biology. His studies provide fundamental insights that are highly relevant to the molecular and cellular mechanisms that we are actively exploring in our center. I believe that this strong scientific alignment makes today's seminar particularly meaningful, as it bridges shared interest in understanding how cells communicate and organize complex biological processes at the molecular level. I hope that today's seminar will not only serve as a scientific lecture, but also as a platform for meaningful knowledge exchange, in-depth scientific discussion, networking, and the development of future collaboration between researchers in Indonesia and our international partners. I would also like to sincerely thank Professor Suetsugu for taking the time to visit Indonesia and share his valuable knowledge with us, as well as all colleagues and the organizing committee who have worked hard to make this event possible. Finally, I hope all participants will find this seminar insightful, inspiring, and scientifically enriching. And I encourage everyone to actively engage in the discussion. Once again, welcome to the Eichmann Research Center for Molecular Biology. I wish you all a productive and fruitful seminar. Thank you very much.
Thank you very much, Ibu Farah. Ladies and gentlemen, we now come to the main scientific session of today's event. To guide us through the lecture and discussion session, we are pleased to have Dr. Yudi Nugraha, researcher at the Eichmann Research Center for Molecular Biology, BRIN, as our moderator. To Dr. Yudi, time is yours.
Thank you very much for the MC that give me the time. Okay, everyone. Good morning. So so this five is too serious for me. I think I would like to firstly introduce you, my sensei. She is my advisor for my PhD at the time. Could you give the slide for CV, Kayla? I think I have a problem with the. Okay, I have a problem with some kind of the internet connection, I think. Yes, it is like, yeah. So Professor Shiro Suitsugu is currently professor in Nara Institute of Science and Technology, Japan. We usually call it NICE. NICE have had an office also in Indonesia in IPB and also UGM, the office for alumni. So Professor Shiro Suitsugu received his PhD and master degree in cell biology for Department of Biochemistry and Biophysics from Graduate School of Science, the University of Tokyo. We call it Todai usually. His research is focused on the cellular plasma membrane and its essential role of distinguishing the inside of and outside of the cell. He and his team focus on the mechanism connecting the membrane to the cytoskeleton and the membrane binding protein connecting the membrane to the intracellular and intercellular signaling for varieties of cellular functional, including proliferation and morphological changes. The group is pioneer in membrane shaping protein of plasma membrane and proposed the mechanism of membrane shaping. So the most important thing is Setsugu Sensei and college contribute to the mechanism of the concept of how cell membrane shaping by the protein having the bar, the name protein is bar, and the bar domain, including the A bar domain, after discovering of WASP, wave, and also ARP2/3, complex mediated actin cytoskeleton regulation. So the time is yours, Sensei. So for you also working with the cell, it is the time to know deeper how the mechanism and how the structure of the cell doing the communication can speak each other. And please ask after Sensei's talk, and then yeah, we really happy to answer. Thank you.
So should I start or okay. So thank you very much for thank you very much for very wonderful setup of this seminar. So I haven't expected that kind of large. So thank you very much, Professor Dr. Indi Damayanti, and also Professor Elizabeth Farah Nobita Couturier, sorry, for a very warm message. And also thank you very much for Ando Harushi Arini for guiding us. So I would like to also thank for a lot of existing collaboration between Japan and BRIN, or also Nara Institute of Science Technology, and here and I hope that continue for the good promotion of your science and our science in future. So for that, I would like to introduce some of our research that we are going. So then maybe I would like to start my sharing of the slides. So please a bit wait. So then today, then maybe my side is also lose the connection now. So how should I go, right? Okay. Okay.
Oh, Bingkes bagus ya.
Now it's coming. Now it's coming. Coming. Yeah. Okay, let's start. Maybe coming. Yeah, indeed. Maybe my lab have 3 Indonesian students now. Then one will join soon. So I like this country. Yeah.
Yeah, maybe the connection is not good. Something wrong with the connection. But yeah, I do better with mine also.
So how should I do? Yeah. Or I can switch to move to just directly connecting screen, but there are a lot of internet audience. Yeah.
Yeah, I did it that way. Maybe from your
This is my
1, 2, 3, 4.
Hmm.
Okay.
Okay, thank you.
Is it connected? Maybe you need to go out first. Then, yeah.
Then now connected. Share the screen again.
Yeah.
Okay.
Okay, but there is no sound there. Highlight. Maybe in the Zoom, in Zoom already sensitive, but maybe the monitor one. Highlight.
Okay, okay. Finally, we can start. Okay, thank you very much for waiting long time. Then first several slides are introduction of the NARA Institute of Science and Technology, and later is only my science. Then I'd like to start this. This is our institute. So then this is maybe representative international student of us. Then maybe I think this institute is not so much famous compared to the other university in Japan, like University of Tokyo, Kyoto University, but we are good place. It's located in Nara and only graduate school only. So dedicated for the fundamental research, mostly information biology and materials, right? Then most famous achievement is IPS research started here. So maybe without this is no IPS in this pluripotent cell in the world, probably. That's something that we are proud of it, right? Then we still are keeping the cutting edge in the Japanese funding framework. So it's not a bad idea to collaborate with us or send some of our young scientists to us. Then this is just edge, large funding framework happening, then we are also part of it. Then in that framework, we are accelerating or emphasizing the collaboration with Southeast Asia. So maybe or keep our institute in your mind, please, right? Then this is our international facilities. Then go back to my research. Maybe screen it doesn't change, but okay. I think on the Zoom, maybe it's already on photo my maybe yeah, it's okay. Yeah, then I need to speak slowly probably. Then this is my lab. This is just my lab only, maybe a lot. Then we have a lot of international students. Then I would like to talk about first bar domains that UD sensei kindly introduced me. That's something fundamental molecular elements, indeed proteins that shape the cell. Then how that kind of things make membrane protrusion. Also, I would like to introduce what is a membrane protrusions. Then what we discovered is membrane protrusions is converted to the vehicle that transfer the materials between cells. That is the cellular vesicles. Then that is going to the cellular delivery of any bioactive molecule of interest. Something that is possibility, then I would like to introduce those possibilities. Okay. Then I want to catch your attention how these kind of studies are important. Maybe one example is cancer. Then this is something experiments of xenograft of tumor cell into mice. Then the mice are injected to tumor cancer cell, then it's grown in mice. Then it can be highly metastatic. I mean, they can migrate well. Then those cells are collected, then culture again, injected again. Those cycles make cell more malignant. The more, moving, more metastatic, right? Then in that case, cell morphology change. That is something for a lot of protrusions, right? Then cellular protrusions are called phyllophodia or lamellipodia. Then those are thought to be such a metastatic apparatus like a foot of humans or wheel of the car, right? Then those are something the consensus of how the cellular protrusions or phyllophodia functions. But we found another meaning of that phyllophodia. That is something source of the extracellular vesicles to communicate the cell between, for example, cancer cell and the surrounding host cell, for example. Those are something the overall framework of this talk, actually. Then I would like to introduce a bit fundamental or more basic. Then this is small g proteins. Small g protein, maybe if you are working on cancer, you may familiar with some cancer-related genes, oncogenes, that are mutated in cancer, that promote cancer progressions. One of that is a small g proteins, and the famous example is RAS GTPases. Then RAS GTPases are mutated, then make a cell proliferate. But these small g proteins are relative of RAS, but it's related to the cellular morphology. Those are called CDC42 or RAC1 or ROA, then called a raw subfamily of small GTPases. Then when we looked at the cell, its cell looks like that, right? Then these are indeed actin filaments. That is something, the framework that support cellular morphology visualized. Indeed, cellular surface is a cellular membrane, but its actin is visualized overall morphology. Then when cell moves or metastasis, then the cell shapes dynamically changes. Maybe I show here. Or I think it should move. Yeah. Okay. Also on the zoom, it moves, right? Then at the edge here, it makes such a fun like things. Then it will move. Then you see something flow of actin cytoscape. It's called retrograde flow, right? Then also you see a lot of actin structures. It's someone is protrusion. It's something bundle. Then it's also make something movement force. That's something basic understanding of cellular protrusion that textbook describes. Then I would like to introduce my old study. Actually, this small GTPases are very found in a very old age already by the Riddling Hall in the UK that first demonstrated this small GTPases, regulate cell shape, especially actin cytoskeleton, Then what is the was the extent to prove that is a microinjection of the protein itself. Protein inserted in very thin needle, then directly microinjected. Then cell morphology change in that way. In case of RAC1 injected, then these kind of fan-like actin filament shape are observed, right? This is my reproduction in my master degree, actually. But when protein are injected, cell shape changes. I reproduce it. Then how much protein can be injected or need to be injected to make such cell change is, of course, precisely measured. Then it's 1 to 10% of the endogenous protein that this cell have. So if a protein have exogenous protein, cells have exogenous protein for the amount of 1 to 10%, then it can be effective. Then that is something that goal of this talk today. I will show you the data that extracellular vesicle can deliver this amount of protein between the cell. So that should be useful to understand how the cell communicates, or maybe we can engineer it to deliver what protein you are interested in. Right? Okay. Then I'd like to introduce my old research. Then how the small GTPase can regulate actin site skeleton is through the WASP or wave protein that UD sensei introduced. So it's already old study. Then, but it's this WASP wave protein can regulate actin site skeleton through the activation of the ARP23 or ARP23 complex. That's something starting of this story, actually. Okay, then a receptor somehow activate RAC or CD42, then it going to wave and ARP3, then actin site skeleton. Okay, then when actin site skeleton reorganize, it makes such a Y branch shape actin filaments. Then at the leading edge of the cell, that cell migrate. I think this is maybe I shouldn't move so much. Zoom slide, maybe. Okay, okay. Then there is something like, okay, maybe there is 5 second lag probably delay. Yeah, okay. Then this kind of leading edge there, then it's something migrating in front of the cell. Then it will have such a Y-shaped actin filaments. This is done by the wave WASP, random oriented. Then this is not randomly oriented, or the Y is facing to the leading edge. Then you see what Y can be made by this, only by this protein. So reconstitution can demonstrate the real molecular network behind the cellular shape, right? Then going back to small GT phases, maybe just to remind you. Okay, sorry, a bit wait. Moving, but maybe just I want to say that this small GT based RAC1 can directly dictate cell migration in that way. Okay. Sorry, I shouldn't put so much movie here, but this moves, then okay, then just a bit wait to go. Then our research question is, how do the cell shape their membranes, build protrusions, and communicate with other cells? Not only metastasis, but metastasis needs a host cell to, or then
After, I think.
yeah, then I think your home may be paid by institute. Then we want to pursue, yeah, such, can I? Or pursue such a fundamental question, how the cell shape is organized, then how that organization change can have a meaning to communicate between the cell. Then after such a vastful wave and acting, we discover so-called bar domains that directly shape the membranes. Then in this cartoon, you may see okay, on the left, you may see a lot of green or red one. Okay, then that will directly shape the membrane, then interact with vastful wave, then insert the actin site skeleton after membrane shape is determined. In that way, the complex cellular shape are made. And what's far on, I think you remember in vitro reconstitutions, actin filament are made randomly. But bar domain can dictate the orientation of actin filament in the manner that fit to the need of the cellular shape, like protrusions or invaginations. Okay. The next is how the protrusion structure are made up of these molecules. This is our book chapter. Then what we found is not only the upstream and the wave here, but also the IVA proteins here. It is not so much eye-catching, sorry. Actin is more eye-catching, but this IVA, the membrane binding protein, that membrane is shaped. Here we say deformed. Then this is IVA. Then IVA is something that example of the proteins that shape the membrane by the shape of proteins, right? Membrane is like Shavon soap, it's soft. Then protein is hard. You know, your hair is a keratin filament is protein is hard. Then IVA is also alpha helix band like keratin filament, it's hard. Then if it's not bind to the membrane follow the shape of IVA. Then the membrane binding surface is convex. Then convex, then it fit to that convex membrane, then it's going to the protrusions. That is what I found in 2006. Okay. Then this is the in vitro reconstitution again to show this element really necessary. Then this is electron microscope picture to show how the riposome is. Riposome is artificial lipid membrane without any protein there. It can be made in vitro in a test tube. Then you see this EM picture. This is like a crushed balloon, right? Then if a protein there, then if IVA or VAR, we are not the first in the VAR. Harvey McMahon in the UK found it first. Then we are following that and then expand the repertoire to be the universal membrane curvature generator. Then if a bar here, then this IVA here, then you see this shape on leftmost is crushed like balloon can be converted to tubules in the middle to FVAR or VAR. Or IVA, it's going to the inside, it's invaginations. Then you should remember that proteins are outside of liposomes. Then it's opposite to the cell. Cell are sphere. Proteins are inside of in the liposome, right? Then FVAR or VAR make tubules in vitro. That means cellular membrane are invaginated or tubulated inward. Then in case of IVA, I means in the inverse of the VAR. It makes protrusions in opposite manner, like bar or F bar can make the membrane tubes inward, right? Then here the membrane are deformed inward in the in vitro reconstitution, I say because proteins are outside the liposomes. Okay, that's what we found with my supervisors long time ago. Then structure basis is that the VAR or F have a concave surface. Then I inverse have a convex surface. So membrane binding surface have a different curvatures. Then a different curvature, I mean plus or minus. Then minus going to the invagination inward. I mean cell membrane have a bending that engulf something inside. Then convex surface, the cellular membrane make a protrusions. Then it make protrusion that I show for the cancer cell metastasis, right? Then when overexpressed, then the proteins are inside of the cell, right? Then it's going to the opposite as in vitro, right? F bar makes invaginations. You see a lot of invaginations. This is a cell, right? Then this is a cell, then it's going inward, right? Okay. Then this IVA, then this is IVA, then it's the protrusion are going outside. Yeah. Okay, understand? Then invagination are going to endocytosis. It's cut into the vesicle, then it's something engulfed. Then IVA is just a protrusions for 20 years, but we found it can be also cut it out to vesicles, then that is extracellular vesicles. Okay. Then convex, invagination, endocytosis. Sorry, concave, invagination, endocytosis. Then convex, then protrusion, then extracellular vesicles. Then in summary, the concave one, I mean F bar or bar are going to endocytosis, but after making these inward tubes. Then outer tubes, protrusions are cut to the extracellular vesicles. That's what we proposed in 2021. So 15 years later after discovery of bar domains, right? Yeah. So change the fundamental idea takes time, actually. Then, but then after this, we switch our research to just shaping or building the cell to more cellular communications by using these vesicles. Then from membrane curvature to cellular protrusion, right? Then how is the membrane shape generated? We know the answer, actin and the bar domains, right? The control, we already know it. It's something RAC1 GTPases or small GTPases is the one with the key regulator. Then convert it to cellular functions. Cellular function may be endocytosis, uptake or something. The extracellular vesicle, protrusion is first is metastasis, food, like wheel of the car, right? But also they have another function. They are released or cut it out, then delivered to the neighbor cell to communicate each other. Like endocytic vesicle, something communicate from outside to inside, right? Then the membrane shape is not simply an outcome of cellular behavior, but it should be an active regulatory one. Then let's make summary. Then IVA is here. Then it's bind membrane like that. Then it make curvature in induction. Then make protrusions. Actin also coming in, right? Then it can be cut into the EVs or extracellular vesicles. Okay. Then going back to this. So when cancer cells are getting malignant, more protrusions are made. So it is easy to understand this is why or for the metastasis. But indeed, not all the cancer cells have protrusions, indeed. Without protrusion, cell can still migrate. It's called amoeboid type migrations. So there should be another meaning. That will be something illustrated here, right? Some dots around the cell. That's extracellular vesicle. Cellular tips or protrusions can be cut it out and left over after cell migrate. Then that already known for long years, but people think this is just something artifact or something that, but it has a meaning to deliver the bioactive molecule between the cell. Okay. Then I will show you, but protrusions, cellular protrusions are never shown to be cut directly to make extracellular vesicles. So we collaborated with Riken group. they have a very high-speed 3D microscope. Then you see these protrusions are moving. I'm not sure on the internet they can see, but some vesicles are released from the tip, right? Then that is directly visualized how the protrusions are cut, or it's really cut, right? Indeed, people don't so much believe that these are something cut. These are understood by the migratory machinery like foot. Then foot never be cut, right? But this can be cut indeed. Okay, then this is the EV, extracellular vesicles. Then we see the extracellular vesicle content by mass spectrometry analysis. I think you may understand that protein can be cataloged by mass spectrometry, what the protein there are, then how much the amount is changed. Okay, it's animated to just to make the EVs, not that super important. Then the result is always shown in such a volcano plot. Then we knock down here. I say mean RNA or MIM. MIM is IVA proteins. Then if MIM is RNA, then it means decreased protein amount, lesser cellular protrusions. Then we collected the extracellular vesicle from these cells and also control cells. Then we see the difference in protein content in the supernatant that contain extracellular vesicles, right? Then we see RAC1, right? Here, RAC1. Then going back to my master study, then, right? RAC1 is something that regulates cell migrations. Then it is released to the outside. Okay. Then RAC1 wave after 3, then actin, right? Then if this RAC1 is in the EV and directly received to the recipient cell, then it should promote cell migration if this protein really functions. That is the next questions for this. Maybe in the illustration, sorry, some part is Japanese, but in the EVs, yellow is the best. Good. Then meme IVA is there, then RAC1 is there. Then this, sorry, screen still not, these two persons. Then the RAC1 content really transmitted between cell. Then if RAC1 is really transmitted, then it should induce lamellipodia and cell migrations. Then if that transmission is in large amount, we should change RAC1 to the protein of interest for such a DDS or cell engineering purpose. Those are 3 next questions from this. Okay, then the effect of receipt on the recipient should be promoting lamellipodia cell motility, right? Then experiment is simple to tackle with this. Okay, coming. Then extracellular vesicles are prepared. I mean, just supernatant are collected. Then those are applied to the recipient cell. But in this case, recipient cell is the same cell type that was used for the collection of the EVs. Okay. Then after applying the EVs, yeah, then you see on the left, no EVs, cell has psyche, no lamellipodia. Then control EVs, some lamellipodia shown. Lamellipodia made by wave protein. Then it's clearly indicate lamellipodia is shown. Then if a meme IVA, protrusion derived EVs are added, then more lamellipodia are shown, right? Okay, then this indicated that this EV can promote cell migration probably by promoting lamellipodia. Okay, then this is some control extension suppression the EV or suppression endocytosis can suppress this effect. Okay. Then how this EV functions, right? There should be two scenarios, just surface binding or endocytosis, and ragam is really transmitted by my microinjection at my master degree. So we did super resolution microscope to trace the behavior of the ragam or cargo content. And then it is really endocytous. The top 3 panels show that. So I didn't go to the details. And this bottom picture show inside ragam appear to be escaped from endosome after they are endocytosed. So typical endocyte route of the EV is endocytosis going to endosome, but endosome is a membrane bag. Then EV need to be escape if cargo is delivered. Then such a escape may be captured by this imaging. So this is a proof that ragam in the EV appear to be directly delivered to the cytoplasm of the recipient cell. Okay. Then going next. Then we then measure the amount of ragam protein that delivered into recipient cell by canonical Western blot. Then in this case, ragam is tagged with hero tag to distinguish the endogenous recipient cell endogenous ragam protein, because the recipient cell of course have ragam, so we need to distinguish the delivered protein from original protein the recipient cell has. Then hero tag addition will change the molecular weight, so we can directly visualize the amount of ragam delivered by hero tag. Then we see clearly the hero tag ragam visible after EGV treatment. Then we made a lot of calculations. Then I didn't go to the details, but amount of the protein delivered by this is similar to or equivalent to the 1% of the total ragam protein that a recipient cell has, right? Then EV extracellular vesicle can carry large amount of protein that can directly induce recipient cell reaction in microinjection levels. Okay. Then we can prove content is delivered in microinjection levels. Then physiology appear to be protrusion and cell migration. Then I would go a bit in vitro or in vivo relevance. Then when cancer cell metastasis, it always go to the blood, right? Then blood have chemoattractant then, right? Then chemoattractant is something called PDGA for growth factors, but those are not only one. Some studies here on the right show that the knockout of that pathway still can induce serum-induced, blood-induced cell migration, but PDGA4 growth factor-induced cell migration is inhibited. So there are still unknown factors to promote cell migrations or metastasis. Then that can be EVs. Then we tested EVs in the bloodstream, however, ragam or not. To prove that, we have several experiments. I think when you culture cell and doing the metastasis analysis, I think you use the fetal bovine serum. Yeah. Then that should have also the EVs, of course. Then those EV can be depleted by ultracentrifugations. Then those cells are treated with the recipient cell with or without endocytic ability. Those are presence of CIP4 or CIP4. Then those are shown to involving the endocytosis of these EVs. Then do the wound healing analysis. Then this shows wound healing analysis. You may not be familiar with wound, but wound is a wound. Like your knife cut your arm, it's wound, right? Then the cellular culture in monolayers. Then it make wound by scratching the monolayer culture. Then you see that this cell here and this cell here. Then in the center is wound. Okay. Then those are closed by this way. Yeah. Then this speed of the closure is the migration speed. Okay. Then in the presence of the serum, FBS, it closed. The knockout the CIP4 and cytic protein, it slows. But in the absence of EVs, the speed are same. So that means the serum EV promotes cell migration through the CIP4 dependent endocytosis. The right bars quantification of this. Okay, then EVs in the serum promote cell migrations. Okay, then indeed we check the RAC1 is really in the EV. It's in the serum. Then we saw a lot of EV RAC1 in the serum in this way. Then we saw also a meme, IVA, is present in the serum. Then here I say LEVs or SEV. I didn't say that so much. Then these are two classes of EVs by separation methods. L is a larger EV, S is smaller EVs. So most of the extracellular vesicle study or EV study are looking SEVs alone. So it a bit LEVs are not so much well noticed before, but those EV having RAC1 more and having mean. Then indeed, adding back this EV can promote cell migration in wound healing. Okay. Movie follows. Maybe on the screen it's not evident, but it induces follower faster movement. Okay. Then also after our study was at a similar time as our study, also another study to make catalog of the EV protein in human serum, so that RAC1 is something prominently included in the human plasma extracellular vesicles. Okay. Then malignancy and the number of protrusions correlated. Then it's not only for metastasis or migrations, but it's also for the EV number. In this kind of series of cells, for the malignancy increase, also the EV number is proportionally reported to be increased by other groups. Then we would like to propose these EVs can be cellular protrusions, directly transmit RAC1 or maybe other molecules, then promote cell migrations in a group of the cell. Okay, then we answered 1 and 2. RAC1 is delivered a lot. Then physiology cell migrations. Then it can be found in serum also. Then it can be involved in cancer cell metastasis. Can be, right? Then I will show you some modifications later on. The number 3, right? In number 3, then to insert protein of interest in this kind of EVs, then we utilize synthetic biology. That the chemical induced dimerization may be already famous. It's established more than 20 years ago. But rapamycin is something binding protein to mTOR. The mTOR is divided into two, FKBP and FRB. Those are fragments. then that are sensitive to rapamycins. Then presence of rapamycin forces dimerization of these two proteins. So with this, MIM-IBA, then Cas12 is a Cas9-like genome editing enzyme. Then these two are coupled by rapamycins. Okay. Then we inserted Cas12 in this way into EVs. And then recipient cell are something implemented with the tricks to see the genome editing delivered. Okay. Then this is something frame shift. You may know that how the Cas12 or Cas9 make gene editing. It's cut, right? Just cut. Then cell try to repair it. But repairment fails sometimes. Nucleotide addition frame shift were made. That's a principal knockout. Then Cas12 target there. Then it's cut. Then frame shift happening. Then after frame shift, GFP is expressed. We insert this kind of cassette into the recipient cell, then see how many percentage of cell can be GFP positive after treatment of the EVs. Okay. Then we see this one. The effect is not that much, but in the presence of rapamycin, we see clearly the increase of GFP positive cell, right? The picture and the graph. Then in this way, we can demonstrate, we successfully demonstrated that any protein of interest can be inserted into the protrusions and can be used for the delivery tool between the cell. Okay. Also, this implies cells are naturally exchange their bioactive molecule proteins each other. So this is something revolution of the understanding of the cell, probably. Okay. Then we can engineer these CVs to insert any protein of interest, ideally. Then we are going back to, or a bit into this more about from the viewpoint of extracellular vesicle research. I think I talked my research based on my research history, cell biology context. But in extracellular vesicle research field, what the impact of this? Then here is the donor cell. Yellow one is EB producing cell. Blue one is recipient cell. Then there are two major sources of the extracellular vesicles. One is plasma membrane derived or protrusion derived, because plasma membrane need to be cut, always it should be protrusions at some extent. Another source is endosome derived. Then endosome have an internal vesicle, it's called multivesicular body, or interluminal vesicles. Then those are stored in the endosome. Then endosome fused to the plasma membrane, inside the vesicle are released. This is called endosome-derived EVs. There's an alternate name, exosomes, right? Exosome in a narrower meaning indicate endosome-derived EV alone. It's something International Society of Extracellular definitions. Okay. Then a number of EVs are larger in endosome-derived EVs. But endosomes are something that protein or anything degradation machinery. So basically, the things in the multivesicular body or endosomes are waste for the cell, right? In contrast, protrusions are facing to cytoplasm. So what they are captured, the protein captured in the vesicle by cellular membrane are not the waste for the cell, donor cell, producing cell, right? Then maybe protrusion-derived EVs are more something fit to the positive meaning for the cell. We shouldn't say the positive or negative for cell because we are not cell, but those are something different, right? Okay. But both things deliver contents to the recipient cell. Then we also this is our review. I mean, to curate various calling of the EVs. I say SEV or LEV in the middle of the talk. Then LEV or SEV, the small EVs or large EV. This is something just a differential centrifugation, experimental-based classifications. Then another EV name is exosome, ectosome, or microvesicles. Those are something origin-related names. Then also another class is molecular marker-based names. Then those are something CD63 or hostatin serine or something. Then those are not mutually exclusive. There are always some overlap. That's something difficult in the field. But at least there you see the CD63 here. Then those are something supposed to be mostly endosome derived. Okay. Then we compare the difference in character between endosome derived EV and protein derived, protrusion derived EVs to see the difference, right? The endosome derived is characterized by the hostatin serine exposure, also CD63 positive, and also the generation protein machine escort dependent. I didn't say that before. Then you may already know that hostatin serine can be used to capture EV specifically. It's commercially available. It is developed in Hanayama Sensei, Kanagawa University. I know him. Then it captures EVs and it's commercially available from Fujifilm Arco. Then we tested whether this technology can capture our EVs. Then indeed, this is that. The PS outside is something the exosome or endosome derived character. Then you see the amount of mean. Then the mean amount is larger for the PS inside. Then I think you may know that phosphatidylserine is not exposed to the surface plasma membrane if our cells are living. When cells are going to apoptosis, their phosphatidylserine exposed. So healthy cell plasma membrane is PS negative for their surface to the outside. Then if you imagine the protrusion are directly cut, it should be phosphatidylserine negative then. Then it's fit to our hypothesis that the protrusion are directly cut. Then also CD63 is mostly negative for the CD. This is flow through and pull down. Then this is CD63 based pull down analysis. Then our EVs are basically protrusion derived EVs are basically negative for CD63. Then this LEV or protrusion-derived EV capable for delivering protein something appear to distinct population of the extracellular vesicle that the people have had a lot of attention before. I skipped the escort, but it's also support the same. Then also we finally put the genome editing enzyme to both. One is CD63 for the representative endosome derived. Then also the IVA coupled genome editing enzyme as usual. Then we compare the delivery efficiency in the middle. Then you see a bit higher for protrusion derived EVs, right? But not just that higher. When you see the proteins inside, then endosome derived EV have more of the proteins compared to the IVA derived EVs. But genome editing efficiency is similar. That means plasma membrane derived or protrusion derived EVs are more efficient in delivering the bioactive molecule within the cell. So that looks suggests the cellular plasma membrane are naturally cut and transferred to the neighbor cell to transmit their content each other, right? Then maybe this is something conclusive slide. Protrusion derived EVs are efficient comparable to microinjections. Then it can be modified. Then it can be also, I didn't say that, but they have a specificity to the target cell. Okay. Then this is summary to catch up. The protrusions are cut it out. Then it going cut it out, travel to the acceptor cell, then transfer the material inside the EVs. It can induce, I say the metastasia alone, but other type of the protrusion-derived EV can induce proliferation, and also related to Alzheimer's disease, probably something. Then also we can engineer this and can induce a genome editing of the acceptor or recipient cell. Then where we are doing in this way is how this protrusion can be made, then how the cargo can be trapped. We engineered, but the natural mechanism to capture the cargo into the protrusion are not clear. And then also we are doing some engineering of it. Okay, these are my collaborators. Thank you very much. Then Terima Kashu, right? Okay, then we have this is my lab now. So I'm happy to take any questions. Thank you very much for kind attentions.
Thank you very much, Sensei, for the very clear explanation. And every slide there's a citation and publication, and very like very clear the research idea and research gap that you answer just now. Here we have we belong to research organization for health. There is so many people working with the cellular, also of course the liver cancer, for example, Mbak Cecil and group. I think also we have also people working with the cellular, including like in vitro assay here with from other research center from beside Eggman. We have also other research center. Is there any question? Maybe before you ask something, maybe it's better also you introduce yourself and also some kind of the short research hypothesis that you know working on. Is there any question? Oh, Mbak Cecil. So Mbak Cecil will ask and maybe you introduce first yourself, Mbak Cecil.
Okay, thank you for the presentation. Sensei is very interesting and new because I learned something new from here. So as mentioned by Yudi, my name is Cecilia. I'm a researcher at the Ekman Research Center for Molecular Biology, Research Organization for Health. So I'm dealing with liver cancer. So we also did some studies on the cargo delivery, but most to exosomes or other EV extracellular vesicles. Regarding this EV derived from the cellular protrusions, okay, so this is something new for me. And then you saw also how the efficiency of the EV derived from cellular protrusion is much better than the conventional, let's say conventional EVs. But we know that between donor cells and the recipient cells, it needs some kind of bidirectional paracrine mechanism. Okay, so so between these two EVs, are there differences how they regulate these communications between donor and recipient of the cells, or it's more or less, I don't know, similar? Thank you.
That's still open questions. Nobody knows so much clearly know it. But when endosomes infected, a lot of endosome-derived exosomes released. That means if a cell cannot degrade, then it can be released out, like garbage point outside. Then that's the kind of regulation can be possible. Maybe cells are under very high stress or something, then inside things are going to out. But it's speculations, right? Then for the protrusion-derived disease, it's something when the protrusion made, it can be increased, like just cellular stimulated by growth factor, it's also induce more protrusion, right? Then in that case, EV number also increased. Then if a cell are getting dense, then lesser need to communicate, then the EV number decreases. But protrusion also decreases at the same time. But those are something correlative analysis, right? Not, reasoning analysis. So then we need to explore more, right? Okay.
Thank you very much.
Good.
Thank you very much, Mbak Cecil, for good question.
Mm.
I think I also curious because it correlates still with Mbak Cecil question.
Mm.
If the cell like the cancer cell like actually EV is very much have a role in that.
Mm.
So you study a lot of the protein like a bar, F bar or something. Maybe I calculate it's like there is 5 protein that you work with this that you explain which the most role in the like doing the metastasis like transferring the EV in cancer cell and then I think what is your hope? I think the engineering for kind of protein to inhibit some kind of protein to inhibit kind of the carcinogenesis or something else with the role of the EV, Sensei.
Then that point, then I didn't talk so much, but EV have homophilicity. I mean, producer cell and recipient cell, if those are cell are same kinds, they can get more. That means if we prepare the EV from cancer cell, then if we inject that EV in the body that having that cancer, that EV is supposed to prefer that cancer compared to the other organs.
Okay.
Then that can maybe specifically target. But of course, it's not 100% specificity. Yeah, but it can be something possible things. Then deliver any protein of interest to the target donor. Yeah, that's
Okay.
one way. One way. Yeah.
Okay.
Mm-hmm.
Is there any question?
Mm.
Oh, Dr. Santi. Dr. Santi is medical doctor working with microbes, right?
And thank you so much, Sensei, for I'm here.
Yeah.
Thanks so much for your explanation. It's very interesting.
Mm.
So I'm working on the bacteria microbes for the antimicrobial resistance and biofilms. So since your presentation, like EV is for the communicating, the biofilm is also have quorum sensing. So is it possible the EV is the recipient is different cell type of bacteria? Is it possible since it's first
Yeah,
time?
it's possible.
Possible. And is it possible for the horizontal transfer gene?
Of course, possible. Yeah, bacteria EVs are also a bit hot field, and people look that for the horizontal transfer
Inside the vesicle cell.
between the bacteria.
Yeah.
Also, but that can be in the gut or something. Then also it can be taken up by the intestinal cell.
Oh, okay.
Then some
Okay.
people say that it can be taken up, then even can go to the brain. But people are still not so much believing that.
So for the EV, for example, from E. coli, EV E. coli 1, and then change to Klebsiella, for example, is it possible different type of bacteria?
Sure. Different type of bacteria EV have different effect, of course.
Thank you so much.
Thank you.
Okay. Clear, right? Oh, thank you. Is there any question more? Oh, Bufina? Or who's first?
Anyone.
Okay.
I-
Yeah, please.
mm.
Maybe you can introduce yourself first.
Okay, thank you, Pak Yudi, for inviting me in this very exciting seminar with Sensei. So, introduce myself. My name is Vina Laksmi from Directorate of Laboratory Facilities, Science and Technology Park. So, my question is, what become the trigger that cause the cell to form a cellular protrusion at a specific location? And also, to what extent does membrane curvature determine whether a protrusion remain on the cell surface or undergoes cleavage and is released as an extracellular vesicles? Thank you.
Sorry, first part of question. Could you
What the initial triggers?
triggers
Yeah, that.
of the EV biogenesis?
Yes.
Maybe it's same as protrusion generations that are something stimuli to the cell, like growth factors, any. If a cell now is that, then whether there are any active mechanism to further push the cut. Maybe we have some candidates, but still something not clear. Then next question, lot of questions, could you repeat again?
Next question is to what extent that the membrane curvature stay as the membrane curvature or cleavage?
Sorry, could you repeat again?
So to what extent does membrane curvature determine whether protrusions remain on the cell surface or undergoes cleavage?
After protrusion,
Yes.
how it can be cut, or
Yeah.
what is the decision usually?
Yeah.
Also, it's the same. It's still an open question now, but something inside the force and outside the force both matters. So interesting example is when people do the exercise means exercise, running or something, EV in the bloodstream increases. So if external force applied to the cell, exosomes or extracellular vesicles certainly increases. So it is believed to induce a positive effect to the whole body. So external force is one of the definite trigger of the EV generations of this kind. Indeed, in our work, force is direct factor to increase EVs. External force is, fluid force. Cell are in the fluid always, or even in the organs are the EV release.
Yeah, is there any question? I still curious, either the protein that you already explained us is determines the structure of complex, and then the molecular dynamic of this protein in case for example like podia going out and lead to move the cell, but also to transfer something doing the work. Did you already work with the structure determination of it, or molecular dynamics?
Tube structure, how the protein can decorate around the tube, it's already well clarified. But how that maybe some defect of the such a line protein there are maybe the reason for the cleavage, or what the things is not clear. But it's something a bit difficult because it's large scale molecular dynamics, right?
Big.
Yeah, very big, right?
Yeah, is there any question? The last question? Oh, still Ria, Mbak Ria, please.
Yeah. Hi, Sensei. My name is Ria from Genetic Engineering. I have question related with your explanation with RAC1 dependent lamellipodia formation. So I would like to ask that whether this RAC1 dependent manner is applicable for all of the cells, even in normal or also in cancer. The RAC1 dependent manner.
Oh, you mean RAC1
Yes.
EV common in all cell types?
Yes, Sensei. Because the one that you show, maybe it is from HEC or CF.
Yeah, it's HEC, right?
HEC. So it is also released by other cells, like from other tissues.
Probably, but we still haven't characterized it yet.
Uh,
But the RAC1 is one of the abundant, except a lot among small GTPs. Its amount is very large. You may know the profilin,
yeah.
actin binding protein, right?
Yes.
The RAC1 amount is similar to that. It's large.
Mm.
So maybe basic idea, larger amount of the protein in the cell are more easy to be released.
Oh.
Yeah.
Okay. So maybe it's does express it.
Yeah,
Oh,
express, yeah.
yes.
Then if a protein have overexpression of some protein, then it can be easily,
Yeah.
spontaneously going to the EVs indeed.
Yeah. Thank you,
Yeah.
Sensei. But maybe a bit beyond of your explanation, but you a bit discuss about the Alzheimer, that maybe this is also applied to the the function of the brain.
Mm.
Maybe communication between cell, it's also happen in the brain, like
Sure.
yeah, neurotransmitter and also synapse.
Mm.
So in this, there is my PhD. Maybe Sensei knows about my PhD. There is some form of the synapse, like philopodia, like mushroom shape of the communication between the cells.
Right.
Do you think this also EVs also
Can.
yeah, exists in this type of
Then some
synapse?
researchers start to say synapses also can be cut it out or even engulfed. It's called trogocytes. It can be cut. Synapses are cut and removed.
Oh,
So it
okay.
can be the EVs also. Right?
EVs also. Oh, okay.
Mm.
Thank you, Sensei.
I think there is a question from Zoom. I think I will read it. Maybe the first question from Nurfianto Giar Pangidung. Good afternoon. I want to ask Professor Sugu regarding the topic of cellular protrusions and extracellular vesicle for delivery of the bioactive molecules between cells. In what form will the medicine or therapy that patient receive be? Thank you. It's mean like if something you can make a
Can be, but no precise idea how it can be. But either if a curvature can be designed, We actually we do a bit on it, but still not so much reach to the efficiency of escaping. And indeed, endosomal escaping is one of the biggest problem in this field. Nobody know it. People know virus can escape from endosome. That's why virus can spread. But equivalent part of the endosome, exosome, is not known. Maybe naturally, endosome shouldn't escape anything. Hopefully. Then there should be something. But people have not so much clearly identified the mechanism behind it.
I think I have a question before I go to the third question. I think this is it's just my imagination. If we cut the eye bar and then it's like reduce the philopodia, it like going up, and in the same times of EV also going down. How we distinguish, is it because of the EV biogenesis in making in our body, and in the other hand it's like it's because of exactly it's from philopodia the movement. So how to distinguish in our experiment? This is not because of the influence of biogenesis in making of the EV.
So your question is, can we separate LEV or protein-derived EV from endosome-derived EV?
Yeah.
Okay. It's not possible in precise saying because we don't have any specific molecular marker that can be pulled down. It's always a mixture of it. Then it would be still not difficult, not easy to distinguish this. Pure EV or something like EV. If you imagine EV like a virus can have a structure. It's very homogeneous, but EV are naturally not. Then there we need more effort to make something clinical like virus particle like pure EV. If we need it's still challenging, right?
Okay, Sensei, maybe the last question from Rizka Yuli Nurfianti. Dear Sensei, I'm Rizka, a PhD student in pharmaceutics regarding a bar domain ability to deform liposome and facilitate endosomal escape. Can we mimic this F bar and I bar structure topography to design smart lipid nanoparticle (LNP) that can artificially trigger of endosomal escape for targeted drug delivery?
Yeah, the same as maybe last answer. So it can be, but still we don't see clear idea to do it. Yeah, virus, but can do it. So we should learn from virus, but virus protein have no homology to any cellular protein.
The same.
Yeah, it's same, right?
Okay, thank you very much. It's like the closing the time, because the time is limited. I think it's like now I'm going to it's still maybe I'm going to just go to the additional question. Maybe one or two question, the last. Okay. Is there any question? If not, I think
Final
Huh?
from Anissa.
there is a question in the chat. Anissa. Is saying like Konnichiwa sensei. My name is Anissa Magvira from Gajah Mada University. I would like to ask about the cessation of the extracellular vesicle from membrane protrusion. I'm curious about how the process of the shading or cutting of EV to different types of the EV. For example, from protrusion of from endosomal also have a different cutting mechanism. Thank you.
Yeah. Of course, it's different because protrusion derived via direct cut from the flash membrane, so its machinery is not clear. Indeed, external force can drive the scission. Then whether we have another mechanism to power enhance that is not clear. But for the endosome derived, it is stored in the endosome. It is clearly shown that escort protein is responsible for it. But escort also still function in plasma membrane for the virus body. But we didn't find any correlations of the escort for the plasma membrane derived DB so far. So something different, maybe. Yeah.
Is there any question from here before we close? Okay, I think it's enough. The time is limited now, so I will give the time to the MC. Thank you very much for a good question and listening our lecture today. I'm really happy and appreciate it.
Thank you very much.
Thank you very much, Dr. Yudi, for leading the session, and also to Professor Shiro Suzugu. And next, we will present a token of appreciation. Please di sini apa di sana? Please move forward. We will present a token of appreciation. Professor Shiro and Dr. Yudi, please come forward.
Okay.
We can open that picture together.
So should I go there? Okay.
Please give a round of applause to Professor Shiro Suzugu for sharing his valuable knowledge. Okay, thank you very much. Okay. Ladies and gentlemen, we have come to the end of today's scientific lecture. We would like to express our sincere appreciation to Professor Shiro Suzugu for sharing his valuable knowledge and insight on cellular protrusion and extracellular vesicles, as well as to Dr. Yudi Nugraha for moderating this scientific lecture and discussion, and to all participants for valuable contribution and participation. We hope that today's scientific lecture will broaden our understanding of the latest developments in molecular and structural biology and further strengthen scientific networking and research collaboration between BRIN and international research institutions. Ladies and gentlemen, thank you for joining with us. Good afternoon and see you at the next scientific event. Wassalamualaikum warahmatullahi wabarakatuh. Kita foto bersama dulu di luar ya, Bapak Ibu.